So the statistic is not too difficult. The first one is just, so the idea is test whether these two are different from one. So the ratio of the two are different from one. And the first test is to use the student's t-test. And that's the formula from your statistic book. And we just use the two number, Ka and Ks, put there. So the numerator is the differences between the two numbers, the two ratios, and the denominator is the variance, the sum of the variance, and take the square root. So that's the student's t-test. So the test of statistics will give you the p-value for you to say whether it's a significant different from the expectation, from the null hypothesis. And on the bottom is another one, we can just use the numbers. So this is like a 2x2 contingence table. So we look at the changes and the number of changes and also the number of no change. So that's also, you put all the total size, that's the NA and NS, and there are two categories. They either change to a different status or they remain unchanged from your sequence comparison. And you're testing whether the synonymous and non-synonymous are somewhat corresponds to each other. So that's the test of 2x2 contingence table, test for independence. So either of the two tests will give you the statistic significance to test whether you can reject the null hypothesis. This is a partial table I copied from another list, is to show the difference between human lineage and chimpanzee lineage. And the short list of the gene here is the one shows like really high Ka-Ks ratio. So the last column on this table is the Ka over Ks for a lot of different genes. And this is, we can look at either lineages. So you can compare the two differences. So it's like the difference between human and chimpanzee. But you can also further dissect whether the evolution is most occur on the human lineage or on the chimpanzee lineage using an algorithm. So that's like the method we usually use. So if we want to look at that, and another thing we mentioned in our textbook is the fast male evolution. So when we look at the evolution rate and higher Ka-Ks ratio, and in some cases we observe certain type of genes show higher Ks ratio compared the rest of gene category across the genome. And one of the example is the male reproductive protein or male reproductive genes. And this is observed in Drosophila as well as in humans. So that's a sort of like an observation we observed, we found in different lineages. So a lot of people would talk about why this is the case, what shaped the evolution of the male reproductive proteins. And another observation people did is the expression differences on the male reproductive proteins. So not only the protein evolution is faster, but also the expression, the evolution at the expression level also faster for those male reproductive proteins. So this is just one of the earlier observations by Swanson using Drosophila as an example. He looked at the DNDS ratio. So the DNDS corresponds to the Ka-Ks we discussed here. Then also illustrate the number, they illustrate the changes. So you can see most of the dots is like below the diagonal. So the diagonal is the equal, like close to 1. But below the diagonal is the ones shows they are really lower Ka than Ks, but there are few data points they show higher Ka-Ks ratio. And in his paper, he also looked at the expression of those proteins between different species lineage, different Drosophila lineages, and demonstrate the rate of evolution and the rate of expression change happened at the same time for these male reproductive proteins.